Section 4. Proteins include a diversity of structures resulting in a wide range of functions. Proteins account for more than 50% of the dry mass of most cells. They're instrumental in almost everything an organism does. Protein functions include structural support, storage, transport, cellular communication, movement, and defense against foreign substances. Humans have tens of thousands of different proteins, each with a specific structure and function. Proteins are the most structurally complex molecules known. Each type of protein has a complex three-dimensional shape. An overview of protein functions, you can see the enzymatic proteins, defensive proteins, storage proteins, transport proteins. The enzymatic proteins, their function is selective acceleration of chemical reactions. An example would be digestive enzymes catalyzing the hydrolysis of bonds in food molecules, such as amylase, digesting starch products. Defensive proteins, their function is to protect against disease. The antibodies inactivate and help destroy viruses and bacteria. Storage proteins, their function is the storage of amino acids. For example, casein, the protein of milk, is the major source of amino acids for baby mammals. Plants have storage proteins in their seeds. Ovalbumin is the protein of egg white, and it's used as an amino acid source for the developing embryo. Transport proteins, their function is to transport substances. For example, hemoglobin, the iron-containing protein of vertebrate blood, transports oxygen from the lungs to the other parts of the body. Other proteins transport molecules across membranes, as shown in the picture. Then we have hormonal proteins, receptor proteins, contractile and motor proteins, as well as structural proteins. Hormonal proteins function to coordinate the organism's activities. For example, insulin is the hormone secreted by the pancreas. It causes other tissues to take up glucose, thus regulating blood sugar concentration. Receptor proteins respond to the cell chemical stimuli. Receptors built into the membrane of nerve cells detect signaling molecules released by other nerve cells. Those receptor proteins are how cells communicate with each other. Contractile and motor proteins, their job is to move. Motor proteins are responsible for the undulations of cilia and flagella. Actin and myosin proteins are responsible for the contraction of muscles. Structural proteins, their function is support. For example, keratin is the protein of hair, horns, feathers, and other skin appendages. Insects and spiders use silk fibers to make their cocoons and webs, respectively. Collagen and elastin proteins provide a fibrous framework in animal connective tissues. Most important, protein enzymes function as catalysts in cells. They regulate the metabolism of the organism by selectively accelerating certain chemical reactions without being consumed. All proteins are unbranched polymers constructed from the same 20 amino acid monomers. Polymers of amino acids are called polypeptides. A protein is a biologically functional molecule that consists of one or more polypeptides folded and coiled into a specific conformation. So a polypeptide is not necessarily a protein. It only becomes a protein when it becomes functional after it's been folded and shaped to do its specific job. Amino acids are the monomers from which proteins are constructed. Amino acids are organic molecules with both carboxyl and amino groups. At the center of an amino acid is an asymmetric carbon atom called the alpha carbon. 4 components are attached to the alpha carbon: a hydrogen atom, a carboxyl group, an amino group, and a variable R group or side chain. Different R groups characterize the 20 different amino acids. An R group may be as simple as a hydrogen atom, as in the amino acid glycine, or it may be a carbon skeleton with various functional groups attached, such as glutamine. The physical and chemical properties of the R group determine the unique characteristics of the particular amino acid. The 20 amino acids of proteins, here we can see the nonpolar and the polar groups, and we can see the acidic and basic. One group of amino acids has a nonpolar R group, which are hydrophobic. So in the amino acid chain, those would repel the other polar amino acids. Another group of amino acids has a polar group, which are hydrophilic. So they would be attracted to water, and they would be repelled by the non negative charge due to the presence of a carboxyl group. Basic R groups have amino groups with positive charges. All amino acids have carboxyl and amino groups. The terms acidic and basic in this context refer only to these groups in the R group position. Amino acids are joined together when a dehydration reaction removes a hydroxyl group from the carboxyl end of one amino acid and a hydrogen atom from the amino group of another. The resulting covalent bond is called a polypeptide bond. Repeating the process over and over creates the polypeptide chain. At one end is an amino acid with a free amino group, which is called the N-terminus. At the other end is called the free carboxyl group, the C-terminus. Polypeptides range in size from a few monomers to thousands. Each polypeptide has a unique linear sequence of amino acids. Here we can see the peptide bond being formed and the water that's coming out. So that's how we get the dehydration reaction. We're taking water away from it. Here you can see the N-terminus, and you can see the carboxyl N-terminus, the C-terminus. Frederick Sanger and his colleagues at Cambridge University determined the amino acid sequence of insulin in the early 1950s. His first Nobel was awarded in 1958, and it was for working out the amino acid sequence of cow insulin in 1952. Sanger was not just the first person to work out insulin's basic structure. He was the first to show that proteins are made of a sequence of amino acids joined together in a chain. Sanger used various protein-digesting enzymes and other catalysts to hydrolyze the insulin at specific places, yielding groups of small fragments. After years of effort, Sanger was able to reconstruct the complete primary structure of insulin. Most of those steps in sequencing a polypeptide have been automated since then. A functional protein consists of one or more polypeptides that are precisely twisted, folded, and coiled up into a unique shape. Because remember, structure determines function. It's the order of amino acids that determines the 3D structure of the protein under normal cellular conditions. A protein's specific structure determines its function. When a cell synthesizes a polypeptide, the chain generally folds spontaneously to assume the functional structure for that protein. The folding is reinforced by a variety of bonds between the parts of the chain, which in turn depend on the sequence of amino acids. Many proteins are globular or round, while others are fibrous in shape. Here we can see the structure of the protein. This is the enzyme lysozyme. So we can see the ribbon model, where we see the beta pleats and the alpha helixes. We have a space-filling model, and then the wireframe model, where you can actually see those carbon structures. In almost every case, the function of a protein depends on its ability to recognize and bind to some other molecule. For example, an antibody binds to a particular foreign substance. Natural signal molecules called endorphins bind to specific receptor proteins on the surface of the brain cell in humans, producing euphoria and relieving pain. Morphine, heroin, and other opiate drugs mimic endorphins because they're similar in shape and can bind to the brain's endorphin receptors. Here we have an antibody binding to a protein from a flu virus. The function of a protein is an emergent property resulting from its specific molecular order. 3 levels of structure, the primary, secondary, and tertiary, organize the folding within a single polypeptide. Quaternary structures arise when 2 or more polypeptides join to form a protein. Here we can see the carboxyl end and the amino end, and a zoomed in picture of the amino acids that make them up. And this is the primary structure of transthyretin. We can see the secondary structure with the alpha helixes and beta pleats, the transthyretin polypeptide, and then the quaternary structure. The primary structure of a protein is its unique sequence of amino acids. Transthyretin is a globular protein found in the blood that transports vitamin A and a particular thyroid hormone throughout the body. Each of the 4 identical polypeptide chains that together make up transthyretin is composed of 127 amino acids. The precise primary structure of each of the polypeptide chains is determined by inherited genetic information, not by the random linking of amino acids. Most proteins have segments of their polypeptide chains repeatedly coiled or folded. These coils and folds are referred to as secondary structure and they result from the hydrogen bonds between the repeating constituents of the polypeptide backbone. spider silk, which is an example of the protein structure. Those would be fibrous proteins. Then the tertiary structure is determined by interactions among various R groups. These interactions include hydrogen bonds between polar and charged areas, ionic bonds between the charged R groups, and hydrophobic interactions, as well as the van der Waals interactions among hydrophobic R groups. These three interactions are relatively weak, but together their effect helps give the protein a unique shape. A strong covalent bond, called a disulfide bridge, can form between the sulfahedral groups, the SHs, of two cysteine monomers, and that acts to rivet parts of the protein together. So with all of these weak bonds, it's very easy to break them apart. Here we can see the diagram of the hydrogen bonds. There's the strong disulfide, the ionic, the van der Waals, and the hydrophobic. And this is all just that polypeptide backbone. The quaternary structure results from the aggregation of two or more polypeptide subunits. For example, globular transthyretin protein is made up of 4 polypeptides. Collagen is a fibrous protein made up of 3 polypeptides that are supercoiled into a larger triple helix. The helix provides structural strength for collagen's role as a connective tissue. Hemoglobin is a globular protein with quaternary structure as well. It consists of 4 polypeptide subunits, 2 alphas, and 2 beta chains. Both types of subunits consist of primarily alpha helical secondary structure. Each subunit has a non-peptide hemic component with an iron atom that binds oxygen. The lack of that iron is what leads to anemia. Even a slight change in the primary structure can affect a protein's conformation and ability to function. The substitution of one amino acid, valine, for the normal one, glutamic acid, at a particular position in the primary structure of hemoglobin, which is the protein that carries the oxygen in the red blood cells, can cause sickle cell disease, which is an inherited blood disorder. The abnormal hemoglobin molecules crystallize, deforming some of the red blood cells into a sickle shape and clogging the capillaries. So here you can see, this picture is in your book, is figure 519. A polypeptide chain with a given amino acid sequence can spontaneously arrange itself into a 3D shape, determined and maintained by the interactions responsible for the secondary and tertiary structure. The folding occurs as the protein is synthesized within the cell. Protein structure also depends on the physical and chemical conditions of the protein's environment. Alterations in pH, salt concentration, temperature, or other factors can unravel or denature a protein. These forces disrupt the weak chemical bonds and interactions within the protein, maintaining its shape. Because it's misshapen, a denatured protein is biologically inactive. We can see that when we add heat to hair or chemicals to hair. If we're curling hair, we can actually change the structure and shape of it. So you're breaking those weak hydrogen bonds. Most proteins can become denatured if they're transferred from an aqueous environment to a nonpolar solvent. Other denaturation agents include chemicals. It can be caused by heat, and some but not all proteins can return to their functional shape after denaturization. This suggests that the information for building specific shape is intrinsic to the protein's primary structure. Biochemists now know that there are proteins that are responsible for folding the proteins themselves. So these chaperonins are responsible for taking in a polypeptide chain and folding it into its individual shapes. They don't specify the final structure, but rather work to segregate and protect the polypeptide while it folds spontaneously. Molecular systems in the cell interact with those chaperonins, marking incorrectly folded proteins for refolding or destruction. Accumulation of incorrectly folded polypeptides is associated with many diseases such as Alzheimer's, Parkinson's, and mad cow disease. Here we can see the diagram. In 2006, Roger Kornberg received the Nobel Prize in Chemistry for determining the structure of RNA polymerase, which plays a crucial role in the expression of genes.